Good morning, everyone. Thank you all for joining our webinar today. I'm Allison Westcott, Customer Service Manager here at ImmunoChemistry Technologies, and I will be moderating your webinar. You may notice that your phone has been muted. However, we do encourage you to ask questions along the way. Simply type your question into the chat dialog at the right and select Send to Host, and your questions will be forwarded to our technical team. We will be posting the video for this recording later this afternoon or tomorrow morning, so look out for an email from me after the webinar with a link to the recording. Now I'd like to introduce our speaker, Dr. Christy Strandberg. Christy is the staff scientist here at ICT. Today she will be presenting on the topic of cell death. And with that, I'll turn it over to Christy.
Well, thanks, Allison, for that lovely introduction. Before I dive into today's webinar topic, I want to take a moment to tell our listeners a little bit about ImmunoChemistry Technologies. We're a privately owned company that was incorporated back in 1994. We're located in Bloomington, Minnesota, which is a suburb of Minneapolis. Now, we began as a company that provided custom assay services. Now, this includes activities like immunoassay design and development, antibody purification and conjugation, and so on. I do want to point out that we continue to offer custom assay services. Our scientists have years of experience with protein chemistry and ELISA optimization, so we can help you develop reliable, sensitive, and specific immunoassays. We can also scale up and manufacture an assay for clients. If you're in need of a service project, feel free to reach out to us, and we would be happy to discuss your project with you. A few years ago, we expanded our company to offer lines of consumable reagents, including ELISA Solutions such as coating buffers, blockers, diluents, and such. A few years down the road from that, we expanded the company by adding a line of products tailored for detecting cell health status, which will be the focus of today's webinar. Now, I also want to briefly touch on our panel of ELISA Solutions before diving into today's webinar topic on cell death. ICT's ELISA Solution product line includes all of the consumable components you need to build a better ELISA. Our coating buffers, blockers, sample and assay diluents, conjugate stabilizers, and wash buffer all work together to minimize the buildup of unwanted proteins to generate a clean signal. These products work together to address common issues associated with specificity, sensitivity, reproducibility, and shelf life. Our goal is to help you develop optimized ELISAs that have high specific signal and low background noise. If you have any questions about ELISA products or services not covered in today's webinar, please feel free to chat them in and I can get you a response after the webinar. There are also a couple previous ELISA-related webinars available to watch through the resources section on our website. You can also contact us by emailing help@immunochemistry.com or by calling us at 1-800-829-3194. As the webinar title implies, today's presentation will focus on ICT's product lines for detecting different cell death pathways. Our cell death detection products include a variety of fluorescent whole cell assays for detecting intracellular apoptosis, necrosis, cell-mediated cytotoxicity, pyroptosis, autophagy, and much more. I would like to emphasize that these kits are not ELISAs. Instead, these kits use cell-permeant fluorescent reagents to analyze whole intact living cells. We also carry numerous products for detection of intracellular enzyme activity, including enzymes such as Caspases and serine proteases. Additionally, we offer products for detecting mitochondrial membrane potential and oxidative stress, as well as other auxiliary stains like propydium iodine and Hooks. Here's a short agenda of today's webinar topic. I'll start by giving a little bit of background information on cell death and the roles of enzymes called Caspases in forms of programmed cell death, such as pyroptosis and apoptosis. I will also introduce our FLICA product line for detecting active Caspases. From there, I'll briefly introduce our audience to some other assays we offer for detecting various cell death pathways. Cell death is typically categorized into non-programmed and programmed cell death pathways. There are several different programmed cell death pathways, some of which will be the focus of today's webinar. Programmed cell death is traditionally broken down into two main categories, non-inflammatory and inflammatory. Apoptosis and autophagy are non-inflammatory cell death pathways, while oncosis and pyroptosis are considered to be inflammatory pathways. Please note that oncosis is not the focus of today's webinar. All of these cell death pathways are important for maintaining homeostasis. Examples include the non-inflammatory normal cell turnover seen during apoptosis, as well as the inflammatory response to infection seen during pyroptosis. Apoptosis involves cellular dismantling that occurs in a manner that avoids eliciting inflammation. Apoptosis features the activation of initiator caspases that activate effector caspases, which then go on to cleave cellular substrates. Apoptotic cells demonstrate cytoplasmic and nuclear condensation, DNA damage and fragmentation, destruction of nuclear proteins on the cytoskeleton, formation of apoptotic bodies, and maintenance of an intact plasma membrane. They also show cross-linking of proteins and exposure of surface molecules targeting intact cell corpses for phagocytosis. Autophagy is also non-inflammatory and features degradation of cellular components within the intact dying cell in autophagic vacuoles. The morphological characteristics of autophagy include vacuolization, degradation of cytoplasmic contents, and slight chromatin condensation. Autophagic cells can also be taken up by phagocytosis. Two examples of pro-inflammatory cell death include necrosis and pyroptosis. Necrosis does not involve activation of a programmed cell death pathway. Now, this is not to be confused with necroptosis, which is a programmed inflammatory cell death pathway and is not covered in today's webinar. The term necrosis has come to mean several things. Pathologists use the term necrosis to designate the presence of dead tissue or cells. Often the term is used to describe accidental cell death that is believed to have occurred in a manner other than a programmed cell death process. However, it should be noted that overall, at its most basic level, necrosis is the sum of changes that have occurred in cells after they have died, regardless of the pre-lethal processes. Necrosis therefore refers to the morphological state seen after a cell has died and reached equilibrium with its surroundings. In the absence of phagocytosis, which is often the case in cell culture-based experiments, apoptotic bodies may lose their integrity and proceed to a secondary or apoptotic necrosis. Pyroptosis also leads to the release of inflammatory contents, but is considered to be a cell death pathway. And this is mediated by the activation of caspase-1, a protease that also activates the inflammatory cytokines IL-1 beta and IL-18. Pyroptosis also features cell lysis and the release of inflammatory cellular contents. I want to circle back and discuss apoptosis in greater detail. Apoptosis is activated through one of two different pathways, the intrinsic pathway or the extrinsic pathway. Both pathways involve activation of initiator and effector caspases. The intrinsic pathway is activated following cellular stress, which results in mitochondrial perturbation and opening of the mitochondrial permeability transition pore. Once the MPT pore is open, pro-apoptotic proteins such as cytochrome C leak out of the mitochondria and into the cytoplasm and activate apoptosis. Once released, cytochrome C binds to the apoptotic protease activating factor 1 or APAF1, triggering the formation of the apoptosome complex. This results in activation of initiator caspase-9. Activated initiator caspase-9 then processes and activates effector caspases 3, 6, and 7. The extrinsic pathway is also known as the death receptor pathway and is activated by patrolling natural killer cells or macrophages when they produce death ligands, which upon binding with death receptors in the target cell membrane induce the extrinsic pathway via activation of pro-caspase-8 to caspase-8. The trimerized receptor ligand complex recruits pro-caspase-8 to the death-induced signaling complex or DISC via the fast-associated death domain or FAD, leading to the rapid activation of caspase-8. Activated initiator caspase-8 then processes and activates effector caspases 3, 6, and 7, which are responsible for the phenotypic changes associated with apoptosis. So what exactly are caspases? Caspases are a family of proteases that serve as critical mediators in apoptosis and pyroptosis. They're named caspases due to their specific protease activity. A cysteine in the enzyme's active site nucleophilically attacks and cleaves the target protein at the C-terminal of an aspartic acid amino acid, which is why the enzymes are called cysteine-dependent aspartic-directed proteases. Caspase substrates are cleaved after a 3 to 5 peptide sequence containing aspartic acid in the P1 position. Mammalian caspases are traditionally broken down into 3 functional groups: initiator, executioner or effector, and inflammatory. The apoptotic initiator caspase group includes caspases 2, 8, and 9, which are found in humans and mice, and caspase 10, which I believe is human only. As their name implies, these initiator caspases are involved in the initiation of the caspase cascade. Apoptotic executioner or effector caspases include caspases 3, 6, and 7, which are found in both humans and mice. These effector caspases are involved in apoptosis and function to dismantle the cell by cleaving various cellular substrates. The inflammatory caspases include caspase 1, which is found in humans and mice, caspases 4 and 5, which are found in humans, and caspase 11, which is found in mice, and caspase 12, which is found in mice and some humans. I want to delve a little deeper into the role of caspase 1 in pyroptosis. As I already mentioned, pyroptosis is a highly inflammatory form of programmed cell death that occurs upon situations such as injury or infection and likely plays a role in the host's antimicrobial response to pathogens. Caspase 1 is activated during pyroptosis by a supramolecular complex called the inflammasome. Unlike apoptosis, cell death by pyroptosis results in plasma membrane rupture and the subsequent release of intracellular contents. This contrasts significantly from apoptosis, where the cellular contents are packaged into apoptotic bodies to be ultimately taken up by phagocytic cells in a non-inflammatory manner. The morphological differences between apoptosis and pyroptosis are highlighted nicely in the image shown on this slide. Note the intact membrane blebbing occurring in the apoptotic cell compared to the ruptured membrane of the pyroptotic cell. This slide shows a simplified depiction of caspase 1 activation by the NLRP3 inflammasome. Caspase 1 is cleaved and activated in response to multiple stimuli. Activated caspase 1 initiates pyroptosis. Activation of caspase 1 leads to the rapid formation of membrane pores that allow for the dissipation of cellular ionic gradients, thus causing swelling and osmotic lysis. Proforms of IL-1 beta and IL-18 are processed by caspase 1 and released. Caspase 1 activity results in the cleavage of chromosomal DNA, but the fragmentation differs from that seen during apoptosis. Nuclear condensation also occurs, but nuclear integrity is maintained. The NLRP3 activation usually requires 2 signals. The first signal begins with the recognition of PAMPs, pathogen-associated molecular patterns, by toll-like receptors. Then, through an interaction with the adapter protein MyD88, this triggers activation of the transcription factor NF-kappa B. Once activated, NF-kappa B is translocated to the nucleus, where it leads to the synthesis of the inactive pro-inflammatory cytokine pro-IL-1 beta. Another potent pro-inflammatory cytokine precursor, pro-IL-18, is constitutively expressed. However, its expression is increased after cellular activation. The second signal is triggered by an ionic perturbation in the cell, such as an efflux of potassium caused by the ATP-dependent activation of the PX2 receptor, which subsequently results in the assembly of the NLRP3 inflammasome, caspase 1 activation, and IL-1 beta and IL-18 secretion. Now I'd like to switch gears and introduce ICT's line of FLICA products, which are designed to detect caspase activity. FLICA stands for Fluorescent-Labeled Inhibitor of Caspases. FLICA are cell-permeant probes that can traverse the hydrophilic lipid bilayer of cells because of their low molecular weight, absence of any charged structure, and their overall hydrophobic character. All FLICA products contain the same general structure, a unique peptide sequence that competes for caspase binding that is flanked by a fluorescent reporter tag such as carboxyfluorescein and a fluoromethyl ketone moiety that irreversibly binds to the active caspase. The upper image shown here shows the contents of our FAM FLICA kit. The lower image shows THP1 cells that were dually stained with the polycaspase FLICA probe, as well as hooks. In the fluorescence microscopy image shown here, only the middle cell is apoptotic, as seen by the green cellular staining indicating caspase activity. The middle cell also has many bright blue spots, indicating that the cell is beginning to die. The cell in the upper right corner is necrotic, no green and scattered blue. And the cell in the lower left corner is alive and not apoptotic. We tell that by the lack of green and the concentrated blue DNA. Before we move on, I want to give our listeners a quick high-level explanation for how FLICA works. When the caspase is in its inactive zymogen form, the enzyme's catalytic sites are blocked. Once processed to its active form, these catalytic sites become exposed. The FMK, or fluoromethyl ketone group on the FLICA probe, is then able to form a covalent bond with the reactive catalytic sites on the active caspase enzyme. This enables the FLICA probes to be retained inside the cell despite any subsequent wash steps. It's important to note that FLICA is not cleaved by the caspase. Once the FMK reactive groups on the FLICA probes form covalent bonds with the two catalytic cysteine groups within each of the caspase reactive sites, that caspase enzyme is rendered essentially inactive. It can no longer cleave any substrates. Here are some sample flow cytometry results. The data on the left is showing our sulfurrhodamine DEVD FLICA. This DEVD amino acid sequence preferentially targets caspases 3 and 7. And on the right is our 660 far-red VAD FLICA. VAD, or VAD, is a sequence recognized by many caspases and is thus considered a polycaspase targeting probe. As I mentioned earlier, our FLICA probes contain the preferred amino acid binding sequence for the caspase of interest. The histograms shown on this slide depict the results from experiments where Jurkat cells were induced to become apoptotic by a 4-hour exposure to staurosporine. After this induction period, cells were incubated with FLICA for 60 minutes, during which the FLICA passively diffuses across the cell membrane where it can then bind to active caspases. Any unbound FLICA is removed during subsequent wash steps, and after the wash steps have been performed, the cells are ready for analysis. As you can see in both instances, treatment with staurosporine induces over 90% of the cell population to initiate apoptosis. Here are some representative fluorescent plate reader and microscopy results. The plate reader data shows Jurkat cells that were non-induced or were induced with staurosporine for 4 hours and then were stained with ICT's FAM-VAD polycaspase FLICA probe. The microscopy results depict staurosporine-induced cells that were stained with our far-red 660 DEVD caspase 3 and 7 FLICA probe. A corresponding DIC image is also shown. As you can see, our FLICA probes work nicely to detect active caspases using a variety of instruments. Here's some data that depicts using flow cytometry to detect caspase-1 activity. ICT's caspase-1 inhibitor reagent, FAM-YVAD-FMK, was used to monitor caspase-1 induction in Jurkat cells that were treated with nigiricin for various periods of time. A common cell pool was spiked with FLICA and divided into separate treatment groups. Starting with the 24-hour samples and then working backwards, 10 micromolar nigiricin was added to the cells and the samples were incubated at 37 degrees throughout the induction process. Following their respective treatment exposure periods, the cells were washed and analyzed on a flow cytometer. The amount of caspase-1 activity detected directly correlated to the duration of the exposure period. The longer the cells were exposed to nigiricin, the larger the proportion of caspase-1 positive cells found in the sample. This slide is showing some confocal microscopy results obtained from colorectal epithelial cells that were infected with a salmonella strain constitutively expressing M. cherry, which fluoresces red. After 9 hours of infection, the cells were then incubated with ICT's green FAM-YVAD FLICA probe for 1 hour in a cell culture growth medium. The cells were then washed and fixed. The confocal image reveals an extruding cell that is infected. There's many red M. cherry labeled salmonella visible within the extruding cell. The infected cells are undergoing pyroptosis as evidenced by the positive staining for active caspase-1. visible as an increased green fluorescence compared to the background levels of fluorescence in the surrounding caspase-1 negative cells. The upper image is a 3D rendered view in the XZ plane and it reveals the cell extrusion. I want to mention our Magic Red product, which also detects caspase-3 and 7 activity. Unlike the FLICA probes, which inhibit the enzyme's activity by covalently binding to the caspase's active site, Magic Red serves as a caspase substrate and can detect caspase activity without inhibiting the caspase. Magic Red is a membrane-permeant probe that is dye substituted with 2 DEVD amino acid target sequences. As a quick reminder, DEVD is the preferred target sequence for caspases 3 and 7. When it's in its dye-substituted form, Magic Red is quenched. However, as this molecule serves as a substrate, as the DEVD groups are cleaved off of the probe by active caspases, there's a corresponding increase in fluorescence activity. Now I'd like to discuss our Necrosis versus Apoptosis kit, which combines our FLICA technology with 7AAD to allow for an enhanced understanding of cell death by simultaneously identifying necrosis and apoptosis. 7AAD, or 7-aminoactinomyosin D, is a red fluorescing live dead stain. It will enter cells with compromised membranes and bind to DNA. It will not enter living cells with intact membranes. We recognize that there is considerable demand for research tools that can allow scientists to determine how cells are dying. For example, when investigating novel drug treatments, it's critical to evaluate biological or chemical compounds for unintended cytotoxic effects on cell cultures. Beyond being able to assess cell death as an experimental outcome, it's extremely important to have some reassurance that your indicator cell population is healthy prior to initiating any experimental protocols. Improper cell handling and cell culture maintenance can lead to unhealthy cell cultures, thus potentially introducing artifacts or leading to difficult to interpret experimental results. A common mistake is to maintain cell cultures at an inappropriate cell density. Long-term maintenance at too low or too high a density can have detrimental effects. The combination of FLICA with 7AAD allows for the simultaneous detection and quantification of 4 distinct cell populations. Any live non-apoptotic cells remain unstained as they do not contain active caspases and have an intact cell membrane, which excludes 7AAD. Cells that have entered early apoptosis will fluoresce green with FLICA as the caspases within the cell are active, but they will not stain red as 7AAD is still excluded by the intact cell membrane. Cells that have entered late apoptosis will fluoresce green and red due to the presence of active caspases and the loss of cell membrane integrity. Dead cells will fluoresce red as they do not contain active caspases and have lost their membrane integrity. This kit is ideal for monitoring cells by flow cytometry. When you plot the FL1 channel by the FL3 channel, you can easily identify these cell populations as they show up in separate quadrants. The lower left quadrant will house the live unstained cells. The lower right quadrant will contain the green staining early apoptotic cells. The upper right section will contain green and red late apoptotic cells, and the upper left section will house necrotic cells or dead cells that stain red only. When we compare mock-treated and staurosporin-treated Jurkat cells that have been duly stained with FLICA and 7AAD, we see that the majority of the mock-treated cells fall into the live unstained category, while very few of the induced cells remain in this category. 46.3% of the staurosporin-treated cells are in early apoptotic stages, and 50.6% are in late apoptosis. The Necrosis versus Apoptosis kit is also compatible with fluorescence microscopy. The bottom of this slide shows Jurkat cells treated with staurosporin for 4 hours were then stained with FLICA and 7AAD. Here you can see early apoptotic cells staining green only, cells in late apoptosis staining green and red, and then necrotic or dead cells that stain red only. As I mentioned earlier, improper cell culturing techniques can lead to unhealthy cell populations. Here, I have intentionally allowed Jurkat cells to become overgrown prior to staining with our Necrosis versus Apoptosis kit. Cells that were maintained at concentrations ranging from 1 times 10 to the fifth to 6 times 10 to the fifth appeared to be healthy. Most of the cells were located in the lower left quarter, if you look at the quadrants. As cultures were allowed to reach cell concentrations of 1 times 10 to the sixth and greater, we see a pronounced increase in the number of cells entering early apoptosis, as evident by the increase in FL1 signal. This graph shows the results from a study where we were monitoring necrosis in response to stressful cell culture conditions. Jurkat cells were transferred to a suboptimal culture environment. This was serum-free DMEM at room temperature and 0.03% CO2. Cell populations were monitored for a total of 3 hours and were stained with a panel of ICT cell viability dyes and then analyzed by flow cytometry. Green live dead stain is represented by the green line. Propidium iodide or PI is shown by a blue line. 7A-AD is shown as a red line, and DRAC7 is represented by the yellow line. Cells that stained positive with each vital dye were considered to be necrotic. I would now like to switch focus to our final topic, the process of autophagy. Autophagy is a conserved intracellular lysosomal degradation process during which cytosolic organelles and materials are enclosed within an isolation membrane to form an autophagosome. The outer membrane of the autophagosome fuses with the lysosome. The sequestered material is subsequently degraded within the autolysosome. Autophagy plays a critical role in maintaining homeostasis by preventing the accumulation of damaged organelles by disassembling unnecessary or dysfunctional cells and cellular components. Autophagy occurs at low levels in the cell under normal conditions and can be rapidly upregulated during times of starvation or stress. Such degradation activities serve to provide nutrients, amino acids, nucleotides, fatty acids, and so on, and energy during periods of elevated bioenergetic demands. ICT's autophagy assay Red contains a proprietary cell-permeant allopathic molecule that fluoresces brightly when inserted into the membranes of autophagosomes and autophagolysosomes. Our kit allows researchers to easily monitor autophagy using a flow cytometer equipped with a green or yellow laser and an appropriate filter. This slide shows the results from an experiment where Jurkat cells were exposed to autophagy-inducing or inhibiting agents and then analyzed by flow cytometry. Jurkat cells were treated with rapamycin, chloroquine, or a combination of rapamycin and chloroquine for 18 hours, then stained, washed, and analyzed. Rapamycin functions as an inducer of autophagy, and it works by inhibiting mTOR, which mimics cellular starvation by blocking signals required for cell growth and proliferation. Chloroquine is an agent that prevents endosomal acidification. It accumulates inside the acidic parts of the cell, including endosomes and lysosomes. This accumulation leads to inhibition of lysosomal enzymes that require an acidic pH and prevents the fusion of endosomes and lysosomes. Chloroquine inhibits autophagy as it raises the lysosomal pH, which leads to an inhibition of both fusion of the autophagosome with the lysosome and lysosomal protein degradation. Although chloroquine is known to inhibit autophagy, in the experimental model I'm showing you here, treatment with chloroquine actually results in an increase in signal, since inhibiting fusion with the lysosome and subsequent lysosomal degradation actually results in a pileup of autophagolysosomes. The combination of rapamycin and chloroquine causes the most significant increase in median fluorescence signal, as rapamycin induces an increase in autophagosome formation and chloroquine prevents the autophagic process from completion. To recap the topics we discussed today, ICT offers a wide array of products for detecting cell health status. We covered apoptotic and pyroptotic cell death pathways, as well as autophagy, necrosis, and cytotoxicity. I would also like to take a moment to remind our listeners that ICT offers a variety of fluorescent probes beyond the ones stated in this webinar today, such as mitochondrial membrane potential detection products, products for detecting oxidative stress, and products for detecting serine protease activation. If you have any questions about other products or services offered by ICT, please contact us by emailing help@immunochemistry.com or by calling us at 1-800-829-3194. As you're planning out your project or working through an experiment, please keep in mind that we have a variety of resources available on our website to help you. You can find webinars such as this one on a wide variety of topics. Just go to the archive section on our webinars page. We also have some short product demonstration videos if you'd like to see some of our more popular kits in action. We periodically update our blog with helpful tips, new citations, and company announcements, so check there for all of your ICT news updates. And finally, we have extensive documentation in the forms of product manuals, safety data sheets, certificates of analysis, all of which are easily accessible from our products pages should you need them. And with that, that's the conclusion of today's webinar. If you have any questions, please feel free to either chat them in or contact us. And thank you for listening.
Well, thank you so much, Christy. That was great. And that's about all the time we have for today. Again, if you had any questions during the webinar, our tech team will follow up with you personally as soon as possible. And as previously mentioned, I will also be sending out the webinar recording within the next day or so. Our next webinar is scheduled for December 17th and will be on the topic of the benefits of sample and assay diluents for ELISAs. You can register for this on our webinar page. If you have any ideas for future webinars, please fill out the Contact Us form on our website, or you can always reach us through social media on Facebook or LinkedIn. Thank you again so much for joining us, and have a great day.